Uno, dai.
Yes. Yes, everybody, good afternoon. Welcome to the afternoon session. We would like to start. We're already a couple of minutes late. For this session this afternoon, I would need your assistance because we have a hybrid session and I need you to help me get through that. There are people that will be talking to you offline, so that means that the presentation has been recorded, and there will also be a person available online. And of course we have people present here physically. So please let's help each other to make it through the session. And Stefano Bellucci from the Laboratori Nazionali di Frascati from Italy to talk about nanomaterials for health science applications. So Stefano, the floor is yours.
Yeah. So in spite of the fact that I'm starting already 5 minutes late, I'd like to take the time anyway to thank the organizers for inviting me in this beautiful city. And in particular, I'd like to thank Professor Stan and Professor Baybarak. So as soon as my slides come in, we will start. In the meantime, I hope you enjoyed your lunch, but not too much, so you won't fall entirely asleep during my talk, which will start soon, I promise. All right. Great, thanks a lot. All right, so, well, that's actually the title of my talk. I'm going to talk essentially about theranostics. So I'm coming from the National Institute of Nuclear Physics in Frascati, which is about 20 kilometers from Rome, and I'm the founder of this group, Next Nanotechnology Group, and I've been for many years dealing and directing different NATO projects. So I don't know if this is working or not, otherwise I'll go like that. Doesn't look like it's working. Oh, all right. So, yeah, I can go like that, no problem. All right. I'd like to show you first my, doesn't work. So I'd like to first show you my place. That's where I'm coming from. It's an institute devoted mostly to high energy physics, but we have technology in it, which is the reason why I'm there. As you can see, my group deals with a lot of different topics, from preparation of materials, especially nanocarbon materials, to the, of course, the characterizations. In my lab, I have spectroscopy, microscopy, and different types of electrochemical characterizations. And the applications are many, but today I will concentrate essentially on sensors, in particular biosensors, as well as I'll tell you a little bit about the toxicity of the materials, which is something that got me involved in the biology part of this about 20 years ago when I started to develop these materials. My worry was about professionally exposed. Then you believe what it is comes into your body through the biological barrier already changes, and it gets surrounded by a biocorona. This biocorona is unstable in the sense that when there is endocytosis, as by chance, the nanoparticle is injected into a cell, absorbed by the cell through a vacuola, thanks to lysosome action, then it gets changed, also the biocorona. And finally, hopefully you have exocytosis, and what you get is completely different from what you put in. Over the years, 20 years ago, we started studying, so here you see just a few results using different types of cell lines to study the toxicity and reactive oxygen species, other types of oxidative stress signal of the cells, and here also some application of a special type of carbon nanotube called bucky paper as a way to heal wounds. So at the end of that work, in 2009, we published a report, a white book in Italian, and then 2 years later, in 2011, was translated into English under the auspices of the National Authority for Occupational Medical Science. And then it was, in 2015, part of this book that I published with my collaborators was taken up by the European Commission to implement regulations and best practices how to use nanomaterials. Now, well, of course, if you want, the idea of nanomedicine can be turned into a positive thing by saying that we want to use our molecular knowledge and our molecular tools in order to improve and preserve human health, which eventually can end up really in the notion of cell medicine as a nanomedicine. Here you see some, well, I just want to stress in these many slides that I will put that nanoparticles can be seen in the lab by a physicist or a chemist, but then can be modified, and then a pharmacologist will see them completely different as a nanovector. A biologist will study them in action in the cells, and then a physician, of course, will see them in living bodies. So we're talking about a very, very complex notion, and because the nanomaterials can be made not only by organic, but also not only by inorganic, but also organic parts, then you have infinitely many possibilities. Here you see some of the functionalization. I took a nanotube just as an example. So here, let's go to more abstract. Here you have a multifunctional nanoparticle. What does it mean? A nanoparticle that can carry different therapeutic means, drug A, drug B, different targeting ways, different targeting molecules, different elements for amplifying the signal, for instance, for imaging, for recognition, and also elements for the stabilization in the blood circle, as well as the targeting moieties to make it more capable of penetrating inside the cell. So here you see an old review 10 years ago of targeted nanodrugs for cancer therapy that we published with my collaborators. Now, well, what's the notion of theranostics? You're all familiar with it. It's kind of a twofold faces, like two coins of the same metal, therapeutics and diagnostics. Of course, they go hand to hand, and it's nice if we can find a device or a technique or a method that can allow us to detect and to apply some therapy. And nanomaterials coupled with different physical means, such as, for instance, magnetic field, can give us such an opportunity to detect by, for instance, magnetic resonance, and then implement a therapeutic route towards curing cancer, for instance, by selected hyperthermia. Okay, here you see some images of this notion implemented concretely in vivo. And okay, I have to go fast because time is pressing. Now, this morning we heard a nice presentation by Fabiana Arduini showing concrete examples how you can work towards realizing, miniaturizing a diagnostic lab, an entire lab, and put it on a chip, thanks to nanotechnology. And a particular sector that I find it interesting over the past few years, for the past many years, has been to implement sensors based on Raman spectroscopy, in particular selected very sensitive Raman spectroscopy. Here you see CERS spectra, and many such examples schematically represented in this slide, and here concretely on a mice. Now, let me get to some examples of what we were able to do about 10 years ago. We started out financed by the NATO organization, Science for Peace program, to realize electrochemical sensing using composite materials. Composite materials incorporated in different types of nanocarbons, carbon nanotubes, as well as graphene, few layer graphene particles. So basically, this is a standard commercial SPE, screen-printed electrode, where on the working electrode we deposit the composite material. You can see that in some cases, for instance, pristine carbon nanotube, here the concentration is 10% in this example for the filler, for the nanomaterial, for the carbon nanomaterial. You can see that there is no effective sensor. In the case of graphene nanoplatelets, top left, or functionalized carbon nanotubes with aminic groups, then the performance of the sensor is rather good, and you can summarize in the area of the cathodic peak versus area of the anodic peak on the bottom right panel. Later, more recently, although already 4 or 5 years ago, instead we dealt with a different type of sensors based on gratings that we were creating ourselves to study the optical properties and the modifications of these optical properties when analyte, small, minuscule amount of analyte deposit themselves over the surface of the sensor. So these are examples. Okay, at this moment you just see the diffraction of white light on different gratings, which the basis is a polymer, and then by some technique I will flash in the next slide, which is holographic technique, we realize grating where the nanoparticles are arranging themselves in an ordered way, in an ordered lattice. These different nanoparticles of silver, lanthanum phosphate, and titania, of course, show different optical properties. Now, here is the method we used by holographic fabrication. This was a collaboration with Germany and Ukraine. After the realization of the device, we remove this glass, and then we have a light source input, a detector to detect the output, and we measure tiny differences in the refraction index of the material when you have the deposition of the analyte on the sensor. Here you see the final result. The dots are the experimental points, different devices, different gratings with different refractive index, and the dashed line is the theoretical prediction based on some method which is called RCWA method. Now, okay, let me move briefly now to part of the activity that we carried out to the therapeutic side. So drug delivery systems, here is the schematics of, well, on the left, sorry for the Italian, on the left you see the reason why you want to have drug delivery mediated by nanovectors, because you want to lower the dose and keep the efficiency and efficacy of the delivery of the drug, just because it is delivered in a targeted way. So you can lower the side effects. In particular, we're interested in those drugs that need to reach the brain. And of course, in this case, you have a barrier, which is the BBB, the blood-brain barrier, which prevents most of the molecules to get to the brain, to protect the brain. So we need to circumvent and bypass, and we thought of using methods related to administration through the nose. Because you can use a twofold. Okay, again, this is just to avoid collateral effects and reach the brain. These are cartoons. So the idea is to exploit axonal transport and transport between the support cells that you see here in the supporting cell in the bottom part of the cross-section of the nasal epithelium. In this sense, this could be an interesting practical way of administering such drugs, because it would, through aerosol administration, for instance, allow the patient to administer the drug to himself. Okay, here is the mechanism of dendrites absorption, axonal pass, and then streaming to the brain. This is just a graphic representation. Now, there is a problem, and that is the mucociliary clearance, which is omnipresent in the nose, because there are the cilia that move in a synchronous way and eliminate the drug towards the rhinopharynx. So we are using, for this reason, mucoadhesive nanovectors to increase the retention time in the nasal cavity. And this is the solution we propose. You can see here some SCM images, not very clear in this, maybe better here. So for instance, just give you one example of the many things we did or we tried to do, focus on the neurodegenerative disease called Parkinson's disease, which is related to a decrease of dopamine in the substantia nigra. So, okay, these are the unfortunate consequences. So we want to we want to deliver the dopamine hydrophilic molecule. And, okay, there is a precursor, which is called L-tyrosine, L-dopa, and then this gives the dopamine. And we would like to, in different conditions of pH, deliver such. Okay, there are drugs, levodopa, carbidopa, but of course their absorption is very, very, very low. So okay, here's the route of the oxidative path of dopamine. I don't have the time to go through the details. So here, the idea is again to go through the nasal cavity to reach the brain, and you can see here the mechanism through which we believe that this will be, we will be able to do that. The idea is again, deliver dopamine, DA means dopamine here, and we have two routes. Today I will show you the polymer dopamine conjugate approach. So bond formation, this is one of the publications we had on this stuff. And okay, so we are using alginate, okay, as a way of encapsulating nanoparticles of alginate, which we had already explored previously for delivery of different type of drugs. And now here you see essentially the characterization UVVs and infrared spectra to select and show the peaks of the dopamine and the conjugate with alginate. You see also the SEM images of these complexes that we created, and the release that we studied in vitro in SNF as well as in PBS to simulate realistic conditions. This is the in vitro study using a specific cell line, SHSY5Y. And so this part can be concluded by saying that the SLM based on a self-emulsifying lipid, for instance, Gelusir, they give us a promising route to increase the DA levels in Parkinsonian rats. We started to work now on Parkinson rats to confirm what we were able to observe in vitro. Now, let me go to an older work, which actually is, I was referring to the different type of drugs that we administer. In this case, we were targeting a rare pediatric disease, which is called pediatric pulmonary hypertension, one of these orphan type of diseases. Here we delivered RNA. Here this is an in vitro characterization, but RNA using as a vector a polymer, actually two different polymers, PEI and PAMAM, with carbon nanotubes, very short carbon nanotubes to increase their biocompatibility. We're talking about 500 nanometers length, multi-wall carbon nanotubes. And okay, this is the TM image that shows the localization of the nanotubes inside vacuola. So this is a concrete example of what I was showing before in a cartoon. And in vivo, we selected the following way, I mean, to administer the complex polymer nanotube drug. Namely, we use a sponge, implanted a sponge in the mices, control on the left, sponge with the complex on the right, and try to, in this case, the disease has to do with angiogenesis, uncontrolled angiogenesis. So we're going, for us, it's important to reduce the number of vessels per field, per optical field, and the average length of these blood vessels as a way to contrast the development of the disease. And in fact, it is quite successful. As you can see, there is a lowering almost of a factor 2. And so that gives us some perspectives for therapeutic angiogenesis and vascular remodeling in general, not only for this rare disease. Now go back to, okay, again, similar to what I showed you before, and a bit similar to what today was discussed in the morning. And all this based on sampling of two-dimensional nanomaterials, so graphene and graphene-related materials, in order to target the pathogen and to be specific through the addition, the functionalization by selective bioreagents. So the aim, of course, is to realize a sensing system that would be compact, low cost, and lightweight, connected, however, and this is important, to an artificial intelligence IoT type of platform that would be able to manage many such sensors in a large environment, for instance, in this building, or in an airplane, or in a supermarket. So this is the scheme of the project that was approved by our government. I'm not going to go through it, but it's wearable. You can use it in a twofold way, either wearable or in air conditioning systems. Now, let's see how, in concrete times, this is a collaboration with the University of Tor Vergata in Rome and the University of Cassino, where I'm also teaching nanotechnology for electrical engineering. So the idea here is the HCOV immunosensor, anti-COV, primary antibody related to the electrode. And here you see, we produce a layer by layer electrochemical construction and then electrochemical characterization of the device that we have. So first, okay, we start, for instance, in this example is multi-wall nanotubes. We started with them because we knew already that they were working very well. Of course, then we extended this to graphene nanoplatelets, and a few layer graphene. So first of all, we immobilize anti-IgG, so secondary antibody. Then we immobilize in phase 3 the secondary antibody, the primary antibody, and then finally in phase 4 we observe the formation of the immunocomplex. Here is the characterization. You can see in the IV characteristics the different phases of the device. Multi-wall, immobilized, secondary antibody, primary antibody, formation of the immunocomplex. It's just cyclic voltammetry, nothing special. And here you see the anodic and cathodic peak ratio, which are the essential features, essential parameters describing our device. I'm not going to go through it, but if there are questions, I will answer. Here you see the optimization of the primary antibody and a first preliminary analytical performance. There are some interesting features to be observed in this dependence of the normalized current in percentage as a function of the secondary antibody. And here you have the normalized current as a function of the antigen. Still in, this is in nanogram per milliliter, this is in microgram per milliliter. So the last, I promised viruses also in food. This is a project that I directed for the Lazio region. Lazio region is the region around Rome. Here is structural funds from the European Union. It was a 2 years project that just finished at the end of 2023 for the detection of foodborne viruses and some prospective, which was not that successful for the removal of these viruses, but what was successful was the detection. And here you see again the same principle, namely a schematic representation of, in this case, more interesting is that we don't use nanotubes, we don't use graphene, we use biochar. Biochar is you can obtain from waste by some pyrolytic, temperature, pressure type of production, of method. And it's interesting because it's a circular economy, green chemistry type of material, which can be as effective, in fact, as some nanocarbon structures. So here we use biochar to decorate the SPE. To our recent